Hi guys, this is chapter 2. We're beginning looking at the cell, and we're going to talk about biomolecules first. So let's start with the carbohydrates. Let's start with just what is a biomolecule? Biomolecules are molecules that are necessary for life. Living organisms make these biomolecules. They are organic, so they have a carbon skeleton, a carbon hydrogen backbone. And organic molecules are made by living things. So living things will synthesize these biomolecules. They are 4 basic types of biomolecules: carbohydrates, lipids, amino acids and proteins, and our nucleotides. So starting with our carbohydrates, I want you to know the monomers here. The monomers are called monosaccharides. So let's remember that the word monomer is one building block. Put together, you can make many of them together. You make a polymer, right? So many of these biomolecules are repeating units of the same type of molecule over and over again. Our carbohydrates work this way, our proteins work this way, and our nucleic acids work this way. The only group of biomolecules that don't are the lipids. So monosaccharides, mono meaning one, saccharide referring to sugar, is the individual building blocks of sugar. And I want you to know the 3 listed here. So glucose is a monosaccharide that is used in our cells, and it's basically the form of energy that is used to make ATP. So this is the pathway that makes ATP. Fructose is another monosaccharide that you can find in fruit, and then galactose is a third. Putting them together in different ways will make different molecules, and the molecules which only have 2 monosaccharides are called disaccharides. So let me just mention that all of these monosaccharides have the same basic chemical formula, C6H12O6. However, they're put together and arranged differently, so they act chemically different, and therefore they are different molecules. If you put 2 monosaccharides together, if you put glucose and fructose together specifically, you're going to get a molecule of sucrose. This is also known as table sugar. Okay, so in your body, when you have sugary foods, your body can enzymatically lyse or hydrolyze this bond and liberating the glucose free and the fructose free. Lactose is another disaccharide that is a combination of galactose and glucose. And what's interesting about people who cannot digest lactose is that they're missing the enzyme, they're not producing it anymore to break this bond, to hydrolyze that bond. If you're not lactose intolerant, then you're still creating the enzyme and you can break the bond, liberating the glucose and galactose. If you're lactose intolerant, what happens is your small intestine, right, where your digestion is supposed to take place, does not digest the lactose. So the lactose moves on to the large intestine. All right, so large intestine has this disaccharide. And this, in large amounts, is not, it's not usually there in large amounts because your small intestine can digest it. But if you're lactose intolerant, the lactose moves to your large intestine, and then the E. coli, or the bacteria that are there, specifically E. coli is one of the more profound lactose fermenters. The E. coli in your large intestine will begin to digest the lactose. It will ferment the lactose. And as a byproduct of fermentation, you're going to get a gas byproduct. And if you're lactose intolerant, one of these symptoms or signs that you have if you accidentally had a lot of lactose is bloating, which is where the gas comes in, and also having that gas, right? You have to expel that gas. Along with that, you have intestinal upset. You might have, diarrhea, where your large intestine is uncomfortable and is increasing peristalsis to move the substances out. The polysaccharide that you want to know is glycogen. So glycogen is our sugar storage. The 2 organs with a lot of glycogen in them is liver and your skeletal muscle. And you can see that we're just repeating our glucose. So every single green dot is glucose. We see a branching pattern. So this is a branched molecule. And every time that your body is low on blood glucose, if you need glucose in the bloodstream, you will just hydrolyze, right? Just break each one of these bonds, and then one of those glucose molecules can go into the bloodstream to be used by your cells, right? You need another glucose, just break that bond again and liberate the free glucose molecule. So this is the way we store glucose in our body. We store it chained up together in a molecule of glycogen. We hydrolyze glycogen to liberate the glucose when we need it. All right, moving on to lipids. So our lipids is going to be, there's different kinds of lipids. The unifying characteristic is that these molecules are hydrophobic, or they have a part of the molecule that is hydrophobic. So the word non-polar here refers to the fact that the covalent bonding within the molecule does not create a charge or a charge difference, so the entire molecule is considered non-polar. Non-polar substances are hydrophobic, so they do not dissolve in water. Okay, and we are concerned with water because we are mostly water, and our chemical reactions and how we deliver things in our body, how we transport things in the bloodstream is also in water. Water is a polar molecule. Okay, so water will dissolve all things that are polar. So polar molecules are hydrophilic. Okay, so there are 5 different classes of lipids. We'll go through each one of these. The triglycerides is a fat. So your triglycerides is composed of a glycerol region and 3 fatty acids. The glycerol is going to be a 3-carbon alcohol. So if we look at this picture here, here's our glycerol. And then we have a fatty acid. So you can see that the fatty acid is just a chain of carbon and hydrogens linked up together. The fatty acids that are in a fat, if you look down here, this is a complete fat or a triglyceride. We have 3 fatty acid tails attached to the glycerol head. The fatty acid tails can be different. So each one of these can be completely different from each other. They can differ in the length, how long the fatty acid tails are, or they can also differ in the number of double bonds. Now, if you have a fatty acid that has all single bonds, like you see here, that is called a saturated fatty acid. A fat, a complete triglyceride with all saturated fatty acid chains would look like that. All of the fatty acid chains are straight. And so when you have a saturated fat, these saturated fats are able to pack very tightly in with each other, right? Because they're basically a very straight molecule. At room temperature, these saturated fats tend to be solid, and animals tend to use saturated fats as the energy storage. So if you think about lard or a steak and you see that fat on the meat or fat on chicken, that is a solid at room temperature, and that tells you it's saturated in nature because it can tightly pack together. Conversely, if you had, let me erase this here. If you had a molecule, which is the glycerol head, and then you have a double bond in the fatty acid tail, as you see here, that double bond will actually bend that fatty acid tail. All right, so here's my double bond. Double bonds can occur, there's 2 ways that double bonds can orient the molecule. The first way, as you see here, is that the 2 hydrogens are on the same side, and this is the cis fatty acid. Or you can see that the hydrogens are on the opposite side, and that's known as a trans fatty acid. You probably heard of trans fats. It turns out that these trans orientation, the trans fats, are very bad for you, for your health, and they tend to lead to cardiovascular disease. So let's get back on track with a unsaturated fat, right? So maybe we have a molecule with 2 areas like this. So you can see that when you have unsaturation points, a double bond in the fat, the entire molecule takes up more space. In fact, we have to give it even more space. All right, so it actually takes up all of this space. So it's very difficult to pack in tightly these unsaturated fatty acids, right? Because they have these shapes to them that take up a lot more space. So at room temperature, unsaturated fats tend to be a liquid, and plants tend to make unsaturated fats to store their energy. So if you think about olive oil, right? Olives being a plant, olive oil at room temperature is a liquid because these molecules can move around each other because there's more space. So that is one clue that you have a saturated versus unsaturated fat at room temperature. If it's solid, it's saturated. If it's liquid, it's unsaturated. There are some plants that make saturated fats like coconut and avocado. All right, moving on to the next. This is a, excuse me, say this is a fat. Okay, ketone, there we go. So ketones are going to be this subgroup of lipids, and they are going to be made from the hydrolysis of triglycerides. What this means, hydrolysis of triglycerides, is breaking down fat in adipose. When you break down fat, you are creating some of these ketones. So the free fatty acids are released into the bloodstream. Some of these free fatty acids are converted to ketones by the liver. These are some examples of ketones, and what's bad about ketones is that if you build up too many of them in your bloodstream, this is called ketosis, and ketones are acids. Here's an acid, and so if you have a lot of them flooding your bloodstream, your blood, you can overload your buffering system of the blood, and your blood can become acidic, and that's called ketoacidosis. This buildup of acetone, which is a ketone, can also give you a breath that smells like alcohol or acetone. So ketoacidosis is very dangerous. You can go into a coma, and this is what very, very low carbohydrate diets risk is when you're starving your cells of carbohydrates, which is what your body uses carbs first, right? Carbs are number one for your body to use. If you limit your carbs, the secondary biomolecule that your body will mine for energy will be fats, okay? So diets that restrict the carbs will then force your body to start breaking down fats. And even though this sounds good, right, it sounds like it's a good way to lose weight, and people do lose weight with diets like the Atkins diet or the keto diet, the danger is that you can build up these ketone bodies, and this can be very bad for you, right? You can get ketosis. All right, these are just some structures. You do not need to know these structures for ketones. Moving on to the phospholipid, all right, our next subgroup of lipids. You can see that this looks similar to a fat. We have our glycerol head. We have two fatty acid tails. And in our third position, we have a large chemical group that contains a phosphate. So this phosphate-containing group, is going to be basically just shorthanded to phosphate head. Okay, so when you see a phospholipid drawn in textbooks, it'll have that hydrophilic head, which contains the phosphate group, and it has your two fatty acid tails. Now, this is an amphipathic molecule, which means that it has both a hydrophilic region as well as a hydrophobic region. So this is a little schizophrenic of a molecule. How does it want to be in water? Half of it wants to dissolve in water, the head, and half of it does not, right, the tails. So if you put it into water, it's going to take on a couple of different forms. It can either form a phospholipid bilayer or a micelle. You can see our bilayer is going to have the heads. There's two layers of phospholipids. The heads will face the water, and the tails will shy away from the water and form a fatty acid interior. This is going to be really important for what can go through a membrane, because the innermost part of your membrane is going to be lipid, right? So let me just write down that these are what membranes are made of. So are molecules that are hydrophilic going to be able to go through your membrane? And the answer is no, right? Because there's going to be a repulsion of hydrophilic molecules moving through a hydrophobic interior. So it's like oil and water not mixing, right? If you have something that dissolves in water, it does not naturally want to go through that membrane. So we have to help polar molecules and help these hydrophilic molecules through with things like channel proteins. Another molecule that can form is a micelle, where it's just a single layer of phospholipids, but you can see that it forms a little bubble, again, with the heads oriented outwards, interacting with the water because it's hydrophilic, the tails shying away. Okay, let's move on to our icosanoids. Our icosanoids are derivatives of a fatty acid, right? So modified fatty acid. And these are really important in communicating between cells and communicating within the cell. The fatty acid it's derived from is called arachidonic acid. And I do mention prostaglandins because this is an important group of molecules that mediates pain and inflammation. So damaged tissue will actually make prostaglandins, and prostaglandins will start a cascade of events that lead to fever or inflammation, and it's involved in tissue damage and repair. This is an example of an icosanoid. So you can see how it does resemble slightly that fatty acid. All right, moving on to steroids. So steroids are a molecule that are derived from cholesterol. These are some examples. These are hormones, estrogen, testosterone, and aldosterone. But let me show you that they all have the same basic shape. So the steroid structure is this 4 fused ring, right? 1, 2, 3, 4. And cholesterol is different from testosterone by their functional groups. Okay, functional groups. So that would be these bells and whistles that are on top of our basic 4 fused ring structure. And your cells can detect these slight differences in functional groups, and they will behave and unused. Their function is going to be very, very different, right? All right, moving on to proteins. So proteins are polymers of amino acids. So amino acids are the monomer. We have, so they're the building blocks. There's 20 different kinds of amino acids. So I like to think of this as the alphabet, right? We have 26 letters of the alphabet, and we have lots of words we can make. And so the same is true for amino acids. There are 20 amino acids we can make, thousands and thousands of different kinds of proteins. So I want to show you the amino acid structure real quick. This is just a blow up. We have our central carbon. We have an amino group here in purple. And then we have a carboxyl group here, which can act like an acid. So that's our amino acid. And then we have an R group, which is going to be the variable here. So everything's going to have a hydrogen on one end. And then the R group is going to be different with the different amino acids. So every amino acid has the same amino group, carboxyl group, and hydrogen. And the thing that's different is the R group. So moving on to look at this is just an example of some R groups. Alanine has that methyl group. Tyrosine has a different ring, glutamate, lysine. So each amino acid is chemically different from each other by that R group. Now, amino acids are linked together by peptide bonds, and the bonds go from the carboxyl group to the amino group of the next one. And there's 2 terms here. So the word peptide refers to small amino, so small chains, about 2 to 50 different amino acids. And so we say peptides are small proteins, but peptides is used. And then proteins refer to just a larger molecule, so about more than 50 amino acids. Let's go quickly over the structure of proteins. The primary structure of a protein is the sequence of amino acids, and the sequence of amino acids is dictated by our genes, our DNA. So you can see we have peptide bonds between the amino acids. And once we have the primary sequence, then the amino acids, or the polypeptide we have made, this long chain, will start to fold on top of itself, right? And the first structures that are created are due to hydrogen bonding. And there's 2. There is an alpha helix, which gives you this curly shape. And then we have a beta-pleated sheet, which gives you a flat region of the protein. So based on these hydrogen bonds, you get either curls or flat areas. And then the tertiary structure is once those secondary structures are in place, then the other chemical bondings between our group come into play. So these are ionic bonds or covalent bonds or van der Waals forces or hydrophilic interactions. So everything else that the chemicals, how they interact, that will create our tertiary structure. So this is a tertiary. It's a three-dimensional structure, and it's pretty much done for most proteins. Most proteins are finished when they reach that three-dimensional shape, and they're ready to go. Some proteins, right, will also go on to need more subunits or other proteins to come together to create a fully functional protein. So the quaternary structure is going to have more than one subunit. So in this picture of hemoglobin, we have two pale pink subunits, and we have two of these brighter red subunits. We have 4 subunits altogether, forming our finished molecule of hemoglobin. Okay, so these are just some illustrations of different protein shapes. Proteins have all different kinds of shapes to them. Collagen is known as a fibrous protein. A fibrous protein is going to be like a fiber, right? If you have a thread, that's a fiber of fabric. And so these are long thread-like proteins. They can be very, very strong. You can see that myosin also has a fibrous portion to it. Then we have the globular protein, which is going to be more round in shape. It has more of a sort of a globby, round, irregular structure. And so myosin has both this fibrous, thin strand, and it has a globular region as well. Excuse me. Moving on to our nucleic acids, which is DNA and RNA. So our nucleotides are going to be our monomers. Excuse me again. Our nucleotides will have 3 parts to them. We have a phosphate group here. We have our carbohydrate, and we have a base. The carbohydrate is going to be different whether you're talking about DNA or RNA. So the carbohydrate in DNA is called deoxyribose. The carbohydrate in RNA is ribose. And the difference is right here, okay? So notice that deoxyribose, we're missing the oxygen group, and so that's why it's called deoxy, and the ribose has it. The bases are in groups they're called pyrimidines and purines, but all of these bases have a nitrogen in them, so oftentimes we call them nitrogenous bases. And the way that they base pair, right, is A to T and C to G. So we can see that A to T, C to G. So our pyrimidines bind to our purines. So we have 2 groups that hydrogen bond together. In RNA, there is no thymine, there is uracil instead, but you can see how chemically similar uracil and thymine are. So let's take a look at the molecules real quick. Oh, this is a cyclic AMP, which we will see this in the future. This is a common messenger molecule that is derived from your nucleotide. All right, so this is overall structure of DNA. We can see how DNA is a double helix, right? That's the word used to describe this double-stranded molecule coiled in a helical shape. We can see that the ends are anti-parallel. We have a 5' end, 3' end, opposite that on the other side. And we can see that what binds our pyrimidines and purines together, our A's, T's, C's, and G's, are hydrogen bonds. So hydrogen bonds hold our 2 strands of DNA together. In RNA, we have a single-stranded molecule. So it's not double-stranded, it's single-stranded. And RNA has uracils instead of thymines. And it's a lot shorter than DNA. And it's functioning as a intermediary molecule that brings the information from DNA to the ribosome to make protein. DNA, of course, is the molecule that stores your hereditary information. Oops, I spelled it. Hereditary. You know what I'm saying. Stores your genetic information. All right. Oops, I already made the marks on this. Okay, so what are the 4 basic types of biomolecules? I have to say that I was looking at this earlier. Oops. And I thought I made the wrong choice, but I made the right choice. Okay. Anyways, it doesn't seem to be liking my pen anymore. So anyways, the question is, what are the 4 basic types of biomolecules? If you look through them, carbohydrates, lipids, proteins, and nucleotides. So that's actually letter A is correct. When I was moving through this earlier, I thought that the word nucleotide should have been nucleic acid, so I thought it was the wrong answer. But in fact, your textbook does consider nucleotides one of the biomolecules. So letter A. All right, so that concludes the first part of our lecture, and we'll move on to the second.